You know, there's a story hidden inside every single one of us. A story that started about 500 million years ago. And it's not about fossils or bones. It's about a family of tiny, super important proteins that basically act as the managers of our bodies. All right, let's dive into this incredible evolutionary tale. So, we're going to be talking about a group of proteins called serpins. Now, you've probably never heard of them, but think of them as the unsung heroes working inside you 24/7. They're the microscopic managers and safety officers, controlling all the really critical systems that keep you going. And I mean, look at their resume. Their job description is just huge. They're involved in everything from making your blood clot when you get a cut, to tamping down inflammation, to even guiding how cells develop. They are absolutely fundamental to our health. So, you've got dozens of these little guys buzzing around, each with a slightly different gig. The big question for scientists was, how do you even begin to make sense of it all? How do you organize this huge, essential family and figure out who's related to who? Well, the first attempt to map them out was pretty brilliant. Researchers looked at their genetic blueprints, specifically at these little patterns in their DNA called introns. And based on those patterns, they proposed a family tree with 6 main branches, which they labeled V1 through V6. But, and there's always a but in science, right? There was a catch. This really neat and tidy family tree was drawn almost entirely using data from mammals. Us, mice, that sort of thing. It's kind of like trying to understand the entire history of cars by only looking at models from the last 20 years. And that led to the crucial question, the one a big 2015 study wanted to answer. Is this 6-branched structure a new thing, something mammals just came up with recently? Or is it way, way older? Would the family tree still look the same if you went all the way back to our most ancient relatives? To figure that out, the researchers needed a time machine. And they kind of found one in the form of the sea lamprey. This creature is what we call a living fossil. Its family line branched away from ours 500 million years ago, so it gives us an incredible window into our deep, deep past. So, armed with all this genetic data, from ancient lampreys to fish to birds to us, the researchers used a really powerful, cutting-edge statistical method. It's called Bayesian phylogeny. Basically, it's a super smart way to reconstruct a family tree from scratch using just genetic clues. And the result? Oh, it was spectacular. Just look at this. This is the new super robust family tree they built. Each color is a different serpent group, and you can see how they all just perfectly cluster together into those original 6 branches. The first map, the one based on introns, it was right all along. The study's author, Abhishek Kumar, put it pretty clearly. He said that their fancy new method completely supported the old classification system. So the big takeaway here is that this organizational structure, it isn't new, it's ancient. Just how ancient are we talking? 500 million years. Let that sink in. This fundamental way of organizing these critical proteins has been locked in place since our last common ancestor with a lamprey. That is just an incredible amount of evolutionary stability. But here's where it gets really interesting. While the organization stayed the same, the family itself grew. A lot. That ancient lamprey, it gets by with fewer than 10 serpins. Us modern humans, we're working with 36. It's a classic story of evolution taking a simple toolkit and expanding it. And this slide illustrates it perfectly. See over on the left where those stars are? That's the lamprey's simple ancestral set of serpins. They're the seeds. Now look to the right. Over hundreds of millions of years, those original genes got copied and tweaked, creating these big, dense clusters of specialized serpins that we have today. So really, this whole thing is about so much more than just putting proteins in the right boxes. What we're really looking at is a crystal clear window into one of evolution's favorite strategies, how it builds complex new things from a very simple starting point. And the process is actually wonderfully elegant. You start with one good ancestral gene, then maybe there's a little copying error, and boom, you have a duplicate. Now, the original copy can keep doing its important job, but the spare copy? It's free to change and mutate over millions of years, eventually developing a totally new, specialized function. The study gives us a fantastic real-world example of this. There's a serpin called AGT. In lampreys, it works as an inhibitor, a stop signal. But over those 500 million years, a copy of that gene in our lineage evolved into something completely different, a hormone that's absolutely essential for regulating our blood pressure. Same ancestor, brand new job, a total career change. Of course, great science doesn't just answer questions. It opens up brand new ones. The researchers spotted something a little weird going on with a group of ray-finned fishes. These fish have strangely small genomes, and they also happen to be the ones that gained new introns. Is there a connection? We don't know yet, but it's a fascinating new mystery to solve. So we've seen this amazing path, right? From a simple starter kit in an ancient ancestor to this hugely complex and specialized system running inside us today. It really leaves you with one final big thought. After 500 million years of building and tinkering, what is evolution building inside of us right now?